Hello, everybody. Thank you. First of all, I would like to thank the organizers for inviting me to this nice meeting. And I would like to present my data, which not contain a lot of bioinformatics, maybe no bioinformatics, but some interesting ideas which may be relevant to biomedicine. So I'm really passionate by chromosomal translocations. Translocations actually and their role in cancer is a very long story. This idea that bizarre chromosomes may be linked to cancer was put forward by German cytologist Theodor Boveri, who didn't live long enough to get his Nobel Prize, which was amply deserved. And actually, he detected in fast-growing cells some bizarre chromosomes, which are called growth stimulatory chromosome. And in fact, these novel chromosomes were translocated chromosomes. But it took actually more than 50 years from the idea to the discovery of actual chromosome, which was done in 1973 by Janet Rowley. So what are translocations? Translocations are a transfer of a segment from one chromosome to a non-homologous chromosome. And if you take a look at cancers, actually many of them are associated with chromosomal translocations. And some cancers, like lymphomas, are all linked to translocations and are caused by translocations. This is the case of most leukemias, of 50% of carcinomas, of a large proportion of sarcomas and myelomas. Okay, so there are thousands and thousands of chromosomal translocations. Some of them are characterized, some of them are linked to specific cancers. Here is the list of some of the known translocations. For example, MYC-IGH translocation, which causes Burkitt lymphoma. There is a database if you're interested. And this database of chromosomal aberrations, which are translocations and deletions, it contains up to now more than 70,000 different translocations linked to chromosomal, linked to different diseases, genetic diseases and cancer. Okay, in this study, we are interested in two specific translocations and two specific diseases. One of them is treatment-induced leukemia, because some people who are treated for primary cancers, such as lung cancer, breast cancer, in more rare cases, glioblastoma, with strong chemotherapeutic drugs, after a treatment, which may be successful, develop secondary leukemias, which are linked to exposure of this chemotherapy. And these leukemias are a disease, a difficult disease in itself, because it is difficult to treat, and it is a great question for chemotherapists to use proper drugs that will not cause the treatment-induced leukemia. Now, these treatment-induced leukemia have recurrent translocations. One of them is 821. We will talk about it later. A second cancer which we are interested in is Burkitt lymphoma. It's a non-Hodgkin lymphoma, which has three forms. An endemic form, which is associated with EBV, a sporadic form, and an HIV-associated form, which is most interesting for us because it's frequent in Europe and USA. And what is interesting, this form occurs against the background of treatment, because 99% of European HIV positive people are treated with antiretroviral drugs, and as the state of the patients improve, but the Burkitt lymphoma frequency does not change, we will somehow want to know, actually, is there any effect of these drugs on the development of Burkitt lymphoma? All right, so what we want to do is to see and to screen drugs that affect chromosomal translocations. But how can we do that in a normal settings? In fact, chromosomal translocations are quite rare, and spontaneous translocations, they appear in about 1 cell in 10 million. And of course, the detection of chromosomal translocation is complicated because there are no clear translocation hotspots. You can detect them by FISH, which is a high throughput mechanism, which has no high throughput mechanism. So we need to develop an experimental system to test drugs that would somehow affect translocation rate. And we decided to put our use a CRISPR-Cas system, and instead of using CRISPR-Cas system to treat something or to relieve mutations, whatever, we decided to generate translocations using this system. How we do this? We use two guide RNAs to target 2 different chromosomes. Then, after a simultaneous induction of CRISPR-Cas and guide RNAs, we get 2 double-strand breaks. And luckily for all of us, these double-strand breaks are repaired to form normal chromosomes in most of the cases, but in some rare cases, in about 1 case in 1,000 to 1 case in 10,000, translocations are formed. Now we can take this system and treat it with specific drugs to see how these drugs prevent formation of chromosomal translocation or increase the probability of this chromosomal translocation. So the principle is induction of 2 DSBs to induce chromosomal translocation. Now we used 2 loci, two couple of loci, one which corresponds to a translocation appearing in the case of treatment-related leukemia, the other translocation which is linked to induction of Burkitt lymphoma. And we did all this in the lymphoblastoid cell line, RPMI 8866. These are just B cells which are immortalized by the presence of Epstein-Barr virus. So ML and D2 are 2 genes that play a crucial role in hematopoiesis. E2 facilitates transcriptional repression, and actually the translocation induces a fusion gene, which is an oncogene. In Burkitt lymphoma, MYC, which is already an oncogene, a proto-oncogene, becomes overexpressed because it's translocated close to the enhancer of the immunoglobulin heavy chain gene, and this promotes oncogenic transformation of B cells. So now we constructed the cell lines which contain Cas9, couple of guide RNAs under a DOX-inducible promoter. We constructed stable constructs, and now if we add doxycycline in this construct, and we made two constructs. One it was which potentially generates MYC IGH translocation, specific Burkitt lymphoma. One that generates AML-ETO translocation, specific for treatment related AML. And when we use doxycycline here, we induce expression of Cas9 quite rapidly for 4 to 8 hours after induction. And this translates in appearance of chromosomal translocations in doxycycline treated cells. These translocations, this is true both for both MYC IGH and AML-ETO translocation. So now, how these translocations are induced? Is the mechanism of translocation induction similar to what you see in normal translocation induction? Basically, translocation are usually double breaks are usually repaired by three mechanisms. One is homologous recombination, which is a fidelity mechanism and which rarely gives chromosomal translocations or mutations. Another is classical non-homologous enjoining, which reunites blunt ends, and another is alternative non-homologous adjoining, which is more error-prone, and this mechanism is the basis of many translocations. Now, we used our system, and we used several inhibitors of different pathways, for example, an inhibitor of homologous recombination, of classical non-homologous enjoining, alternative enjoining. And what we see actually that when we inhibit classical non-homologous enjoining, we have a huge increase in the induction of translocations. And when we inhibit alternative non-homologous enjoining, the number of translocations decrease. So this means that in our system, chromosomal translocation are induced by alternative non-homologous enjoining as in the normal system. Now we decided to test different drugs that drugs that are used to treat different cancers for their capacity to induce induce translocations in our system. And here we use a system of treatment related AML. So we tested for the capacity to induce 821 translocation, AML-ETO translocation. You see that these drugs have different effect on cell viability, but most of these drugs do not affect the translocation rate, except with methotrexate, which was found to stimulate chromosomal translocations, and carboplatin and cytarabine. They also produce some positive effects, although they are not statistically significant. So the conclusion is that some anti-cancer drugs indeed increase the frequency of T821 translocation, and these drugs actually have already been implicated in oncogenesis, because it was known that methotrexate treatment for some other diseases induces cancer in some patients. But what is interesting is that this effect is specific for AML, because when we try to treat B cells, with B cells that contain that produce 814 translocation specific for Burkitt lymphoma, we don't see any pronounced effect of the drugs on formation of of Burkitt lymphoma, specific translocation. And for as soon as we know, there is no effect of treatment on, and there are no cases of secondary Burkitt lymphoma. Now, the next test was for the test of antiviral drugs that are used to treat HIV positive people. The question was whether they affect somehow formation of chromosomal translocations, or maybe there are some drugs that can prevent chromosomal translocations. Well, most of them are quite natural relative to the translocation rate. But we found some drugs, for example, Bacavir or TAC-779 or others, which increase the frequency of chromosomal translocation, which actually means that the prolonged use of these drugs may potentially increase the frequency of Burkitt lymphoma. So we'll have to provide this information to doctors for consideration. Mm-hmm. So to conclude, that we developed 2 experimental human cell models to study translocations. These models reproduce the generation of specific DSBs and form the translocation. And we have, using this system to screen drugs that are used as chemotherapy and antiretroviral therapy, we identified several drugs which include methotrexate, abacavir, emicitrabine, and TAC-779, which may negatively affect, should promote formation of chromosome translocation. And our system can further be used for high throughput screening of drugs that potentially induce or inhibit chromosomal translocation. So this system is actually quite versatile. What can we do with this system? We can detect the efficiency of genome editing using a one-step procedure. This was published recently. And then we can study the mechanisms of chromosomal translocation, asking ourselves why different translocations occur in different cells, how and do oncogenic viruses affect the formation of chromosomal translocation, or what happens in cells immediately after a chromosomal translocation event. I'll stop at that because we're out of time. And I would like to thank my collaborators. This work was done mostly by Anna and Diego. And this work was supported by many agencies, which I will also thank. It was done chromatin dynamics and metabolism, Cancer Lab at Gustave Roussy Institute. But before I finish, I would like to invite you all to a meeting on the cell nucleus, which I'm organizing next year in Prague, Czech Republic, and taking into account recent situation. Actually, we welcome scientists from Russia to participate in the meeting, and we will make everything so that you will be able to attend if you want. So thank you for your attention, and if you have a question, you can address it to me directly.